Welcome to The Explainer. We're jumping right in today to tackle one of the biggest mysteries in human biology. Why do our minds seem to slow down as we get older? We're going to look at what some call the brain's garbage problem and what some incredible new science is telling us about it.
You know, it's something we all think about. Why does our thinking feel a little less sharp as the years pile on? Well, for decades, scientists have been chasing this exact mystery. And now, a pretty groundbreaking study is revealing some amazing clues.
And it turns out the answer might be hiding in a really unexpected place: the old, leftover proteins, basically the garbage that our brain cells produce. This new research gives us a whole new theory by looking at what happens when the brain's own cleanup service starts to break down.
All right, let's put on our detective hats for this one. For years, in the cold case of brain aging, the prime suspect has always been the same thing: this buildup of old, broken-down proteins that just hang around and cause trouble.
But here's the thing. The investigators, the scientists, they faced a huge problem. Inside a living, functioning brain, how in the world do you tell a brand new protein from one that's been sitting there for a while? It was basically impossible to watch the process happen in real time.
So, to crack the case, they needed a new kind of forensic tool. And that's what they built. They engineered this revolutionary way to tag proteins the second they're made, giving them this incredible, unprecedented view of the whole crime scene as it unfolds.
It's called BondCat, and you can think of it like a special kind of molecular dye, a highlighter that only sticks to brand new proteins. This means scientists can follow their entire life story. They can track exactly how long they last and where they end up. It's a total game changer.
And armed with this powerful new tool, they found their first major clue, and it's a big one. It turns out, in an aging brain, some proteins are seriously overstaying their welcome.
Get this, what they found was absolutely staggering. In an aged brain, that whole protein recycling system slows down so much that it takes almost twice as long to clear out old proteins compared to a young brain. Twice as long!
I mean, the difference is just night and day. In a young brain, protein cleanup is this efficient, well-oiled machine. But in an older brain, that same process just grinds to a halt. Proteins that should have been thrown out a long time ago just end up lingering.
So that leads to the next obvious question, right? What exactly are these old proteins that are just hanging around? And what are they doing in there? Well, the scientists found that these lingering proteins tend to get sticky and clump together. So they created a catalog of every single protein involved in these clumps, and they call it the agrigome. You can think of it as the brain's most unwanted list for protein squatters.
And the scale of this problem is just mind-boggling. We are not talking about just one or two bad actors here. The list has over 1,700 different proteins that are clumping up inside aging neurons.
So what's actually on this most unwanted list? Well, a huge chunk of them are synaptic proteins, the very ones that are essential for our brain cells to talk to each other. Some are already well-known troublemakers in brain diseases, but a lot of them had never been linked to neurodegeneration before. But here's the real kicker. A whopping 69% of these clumping proteins are directly linked to genetic risk factors for brain disorders.
So just when it seemed like the case was pointing directly at these misbehaving proteins, the investigation took this fascinating turn. It turns out there might be a surprising accomplice at the scene.
Okay, so let's meet the microglia. These are the brain's dedicated immune cells. They're basically the cleanup crew. Their job is to patrol the brain, get rid of all the waste, and generally keep things neat and tidy.
But here's the twist. The scientists looked inside the microglia from older brains, and they found them stuffed with those exact same unwanted neuronal proteins. They were especially full of synaptic proteins. The cleanup crew was getting clogged with the very junk it was supposed to be taking out.
And that leads to a really startling implication. What if the microglia, totally overwhelmed by the sheer amount of junk proteins, are resorting to a much more drastic measure? Instead of picking up trash piece by piece, they might just be bulldozing the entire neighborhood by swallowing whole synapses, the brain's vital communication hubs.
So what does all this new evidence, this new theory of the crime, if you will, what does it all mean for our understanding of brain aging and what we can actually do about it?
Well, it gives us a really clear 5-step chain of events, like dominoes falling. First, protein recycling slows way down. That leads to old proteins clumping up, especially at the synapse. Then, the cleanup crew, the microglia, get completely overwhelmed by the mess. So, in a desperate move, they might start destroying entire communication hubs. And this loss of synapses, that could be a direct cause of cognitive decline.
And this leaves us with a really provocative and actually a pretty hopeful question. If a faulty garbage disposal system is the root of the problem, could boosting it, helping the brain take out its own trash more efficiently, be the key to protecting our minds as we age? It's a fascinating new clue in this ongoing quest for lasting brain health.